Okay, so hi everyone. This is the QPAD Organized Point Count Data Analysis Workshop, and I'm Peter Salimos, and I'm going to explain what QPAD is and how that might be useful for your work in analyzing point count data. And if you're wondering, QPAD is not the latest Apple device. You'll learn a lot about it and how that all the letters, what those mean and how that comes together. So camera on is optional when we open up for exercises or just discussion. Feel free to turn it on, but if you don't want your face showed because we are recording the meeting, it is optional, totally up to you. Go for a break as needed. We're going to have 2 breaks for sure, so that I can also get up and move around a bit. But just whenever you are in a class or in a meeting and you would otherwise in person would just go out for a few minutes, it's the same here. Zoom is not different. You are not tied to your chair. Go around, move, stretch, have a drink. So feel free to do that. I'm also not going to see every one of you on my little tiny screen. And if you have questions, you can ask them live or put that in the chat. Juan Andres is going to monitor the chat. And if you, like many of you, have similar questions, we might just collect them together so that we can come back to it after the break or when we are having those kinds of discussions. So thanks, Juan Andres, for doing that. And a little bit about me for those of you who don't know me. I'm a statistical ecologist, started as a biologist, but then turned more to the quantitative side of things. And as a result of that, I wrote many R packages or contributed to these. A few of these are mentioned here, like Vegan Detect, which we are going to use a lot today. Resource selection, and my most often downloaded package, which has nothing to do with science, PB Apply, that just draws a progress bar, but people love it, although Detect, in my opinion, is much more useful, and that is going to allow us to estimate many important aspects of bird ecology. Sometimes I teach like today, and you can reach me through my email that you've known because we contacted each other, or on Twitter at the psalimos hashtag. And I wanted to note that I'm situated on indigenous land in Treaty 6 territory, land occupied, traveled, and cared for by indigenous people since time immemorial to the present day. So if you're in a different treaty territory, just substitute yours and those indigenous peoples and try to think about this while you see this slide. And so in this course, you're going to learn about how to analyze point count data, and not just like a very specific project, because that is usually well thought out and you have a specific design that meets your expectations about what you want to study, how you want to study it. But when it comes to putting together different projects that might have collected the data under different protocols or maybe using different technologies, how do you combine these together? So this might not just go for bird point counts, but for many different things like amphibian surveys or camera traps. I'm going to use bird examples, but just think about how it might apply to your specific data set if that happens to be not bird data. And then, because we are going to deal with statistical methods, and all of those come with assumptions, either stated or not even known. And we're going to explore many of the assumptions involved in point count data analysis. And we're also going to look at how to violate this, or does it matter if we violate these assumptions? Which assumptions are more important to care for, and so on. So today, after this introduction, kind of putting everything in context, why we should care, we're going to look at the data, how we are going to manipulate the data in the coming days. And then I'm going to do a little bit of primer in regression techniques, which I'm pretty sure you're all familiar with. I just wanted to lay out this basis that we are going to build on in the next days. So if there's any concepts, we are going to cover that today. And if this is not new to you, that's still, I think, going to be helpful to have a refresher. If it's new to you, then feel free to stop me and ask questions. On Thursday this week, we are going to talk about the biological reality out there that we're trying to capture and model. On day 3, we are going to look at the detection process. Once there's an observer who is collecting this data, what kind of processes are influencing those, and then a little bit talking about recordings, which is if you as a human observer is not directly observing things in the field, but just put out the device, and then later on you listen to these recordings or analyze it in some other way, how to put this together with human-based bird data. And then on day 4, we are putting these together and look at how to account for the various types of biases, either due to bird behavior or the observation process, so that we can analyze data that might be coming from different sources. And also, as time allows, we look at some complexities, especially roadside surveys, because those are quite prevalent in a bird point count data world. So everything that we need, you can install it using this line. And I think many of you have done this before today. If you haven't done that, you can just do it now while I'm talking. And if you run into any issues, few people couldn't install all the packages from GitHub. If you go to the course repository, you're going to find a PKG directory where I put the package sources, so you can download those zip files and install them locally if that's something that you need. And also Juan Andres will be able to guide you in that. And now, how to get the course material that we are going to need after my intro talk. So if you go to this github.com/bsalimas QPAD-workshop/releases, you're going to find a zip file for day 1. If you save that file, then you will be able to just unzip and you'll see QPAD workshop day 1. And this is the slide I'm using now. If you scroll down, see this workshop.rproj, our project file, double click on it. I have it open, but I'm just going to open it again. Then this should open up the project in RStudio. If you are not using RStudio, you should still be able to use everything here using your preferred GUI for R or command line. Just make sure you set the working directory to the folder, this QPAD workshop day 1, and you will be able to access those R markdown files. For example, the next one is going to be data processing. If you click on it, then you see the code, and then we can go from there. One other thing I wanted to mention is, so every day I'm going to have maybe some updates, not necessarily for the days before, but for the next day. So if you're using Git or just downloading another zip file and you still want to edit these files, add your notes to it, and keep it separate from what I'm going to provide so that your edits won't get overwritten by what I'm providing you on the next day, then you can source these functions and then say QPAD local day 1. Then I see files copied, work in your local copies. Now, among the files, you're going to see all the day 1 files copied over. So now, if I were you, I would work in these. And then whatever you do, you can just copy these files after the 4 days into the same place. So you have all of those files together. So this is not tracked by version control. None of this is going to get overwritten. So we'll come back to this. Now let's go back to the slides. So we talked about this local copy. This is optional, totally up to you. This was just one way I thought I could make this easier, because we are going to update this course material over the 4 days. So that was my little introduction, housekeeping, and how to get what we need for today. Any questions so far? All good? All right. So we often hear apples and oranges, and we shouldn't really compare them. And if I look it up on Wikipedia, a comparison of apples and oranges occurs when 2 items or groups of items are compared that cannot be practically compared. So this happens when you want to combine bird point counts with whale surveys, for example. It's not really easy, although both might use distance sampling. So this is not what we are talking about here. But we need to recognize that how we measure things can have big impact on our results. So that whale example was just maybe a slightly similar way of collecting data, but totally different ecosystems and organisms. Sometimes the ecosystems and the organisms might be the same, but I can say I saw 5 magpies walking down the street. Often we don't even mention how we collected the data. We just say, this is the outcome, 5 magpies. You might say, I only saw 1, but you happen to sit on your deck. So that is a smaller space, a different environment, while I was moving. And like today's garbage day, magpies are everywhere picking the trash. So there are all kinds of things that might come into the picture when we try to compare our data. And these are the types of differences that we're trying to account for in our analysis, putting together different data sets. So we're more likely to compare apples to apples, a delicious to a Granny Smith. So we need to think about what effort went into the data collection. What was the area? What was the physical space that you searched while you were looking for species or specimens? What amount of time you spent? And sometimes, as a measure of effort, people can talk about the number of individuals identified. So how, for example, the number of species is changing as you have more and more individuals. That might be familiar from diversity, community ecology. And also the observer's experience and skill level might come into the equation as well. For example, a good proxy might be number of years spent in field work, or what is your hearing ability, eyesight, which might relate to age, unfortunately. Or if you're using technology, is the microphone sensitive enough? Are those calibrated? Was it damaged? And so on. So the goal is to make our measurements comparable. And there are different ways of doing that. But so why should we do that might be the first question. Because these effects can be significant. So for bird point counts, Steve Matsuoka and coworkers, including me, looked at many species across Canada and Alaska and a couple of northern US states, and looked at how the different point counts compared to each other. For example, if you are surveying a 50-meter radius area versus unlimited counts, you can see the white and the gray, how the same counts using different methodologies. So when you have these things nested inside, you can look at just a 50-meter part and then 50 plus everything outside of it. And if you look at these, then you see the average counts are, there's a 2 to 3-fold difference. If you look at the count period, whether you count up to 3 minutes, 5 minutes, or 10 minutes, you're going to see slightly less, but still a consistent increase. And for the unlimited counts, it's even higher. And these error bars just indicate that you probably shouldn't really ignore these differences. So if one project was done 3-minute unlimited, then you're going to get this. If the other method was done 10-minute 50-meter, you're going to get this, even though the abundances and the counts, the abundances might be exactly the same. And if you're unlucky and these projects happen to correlate with some environmental gradient or habitat types, then you're going to attribute these differences to the wrong factor. So maybe it is not really land cover that is driving the differences, but methodology. So you need to be careful because you see all these huge changes. Now let's define what a point count is. When we talk about point counts, we imagine that the trained observer who knows birds goes out into the field, records all the birds seen and heard from a point count station, which is a given location, and does this for a set period of time, and takes notice of the bird individuals within a defined distance radius. And depending on methodology, you might want to put the individuals into different distance bins or time intervals. So if one was detected close to you in the first 3 minutes, you would put it down as like that. And usually we want to answer the following questions. How many individuals are out there? So what is the abundance? What is the population density? Is this location part of the species range, which is their occupancy? Can I find it or not? But I'm not really interested in how many. How is abundance changing in space? So how can we best describe the distribution as a function of let long, land cover types, and so on? How is abundance changing in time, which is the trend? We often are interested in all of this, and trend is used most often in conservation ranking of species, but more often abundance, or especially for rare species, that is also a quite a useful and important metric. And also, you might not be interested in trend or abundance over large spatial or temporal scales, but you might ask, what is the effect of a treatment on the relative abundance of the species? So for example, if you are to develop an area that used to be some kind of a woodland, and you want to create a parking lot there, what kind of effect that might have? So compared to the expected abundance in a forest, how much lower or higher abundance you would get in a developed suburban area. So these are the types of questions I could think of, or that these methods that we are going to discuss can be used for. So now, if you want to somehow standardize your data, then you can look at how to standardize it by design. And by design, I mean, if you haven't decided yet, maybe you should pick a methodology that would allow you to compare it to other data sets. So just go with what other people are doing, and that way your design is going to match theirs. And And also, when you're thinking about design-based standardization, then you really want to find this optimal trade-off between maximizing the efficiency so that you can count the most amounts of bird species within the least amount of effort. And so how to spend as little as possible to achieve all your objectives. And so as a result of these thinkings, people came up with suggestions. Please follow these standards so we can compare data across many projects. And we put a lot of thinking into this, and this is just going to minimize time spent at one place versus the kind of community that you can count in early mornings, for example, for BBS, breeding bird surveys. But when we, in the same paper of Matsuoka et al., looked at how often people followed these recommendations, we found that only 3% were actually doing what was recommended in the 90s. And so this is all good. This is nothing wrong with people not following standards, but we kind of asked the question why this has happened. And we thought that this coincided with a couple of other things. For example, statistical methods to account for these differences became more accessible to people. So nice software now are available. And that just meant they weren't thinking that much about the design because people assume that stats is just going to solve everything. And that is certainly the case to some extent, but also not always working. This is just a map to show how you can get different kinds of methodologies across northern North America, and all the colors indicate a different method. Like the fill, the outline, those refer to the area sampled and the time intervals included. You can see this is a really colorful map. That means there are all kinds of methods out there. And as part of the Boreal Avian Modeling Project, we're trying to normalize and standardize these data. And that's kind of the root of what I'm going to talk about, this QPAD approach, how to best do this. So this is an issue when you look at large spatial scales and many data sets. And this is when you start thinking about how to best achieve this. And also just the size of the data kind of makes you wonder whether you should go full on Bayesian and use Markov chain Monte Carlo and wait months to have one model converge, or you should just find something that runs quicker and still gives you 99% of the same results that you would otherwise get. So these, I wouldn't consider these shortcuts, more just smart solutions. So now you can think about how protocols can differ when we talk about point count data, or what drives this variation among projects, or just why we have abundant following protocols, which I kind of answered. And I'm not opening up for suggestions now because this day is really packed and we need to get through a lot of things. But just these questions come to your mind like, okay, so what is driving these differences? So one reason why, was someone, sorry, go ahead. Okay, then I'll go ahead. So you get different detection probabilities, and what we mean by that, we are going to talk about it in length. So detection probability is when something is there to be detected, whether you detect it or not, like a bird species, whether you see or hear it when it's there. And that probability varies. We know very well with time of day, time of year. So even if you spend the same amount of effort and you search the same area, detection might vary. So you can't really control for everything by design unless you have a really restrictive protocol. And also, there's difference across programs. One might be looking at trend. The other one might be looking at occupancy. And these different goals and constraints, just a budget, what kind of training they have done, how they can access the sites. Is it like you can travel around because there are roads, or you need quads or helicopter access? Those are all going to decide or factor into what you can and cannot do or easily do. So that's why people started moving away from standards. And also, there are all these model-based approaches out there for unmarked populations, which means we are not marking individuals so that we go back later and see how many of those marked individuals we can find, but purely based on unmarked population surveys. We can use many of these, like double observer, distance sampling, removal sampling, multiple visit occupancy or abundance models. A couple of papers linked here, but there are many more. And what we are going to focus on during these 4 days is removal sampling and distance sampling. So these models have come to the forefront lately in the early 2000s. And since then, it is really hard to publish a paper without some kind of consideration of detection error. But all of these models come with assumptions. The most important assumption that we usually think about when we talk about birds is this population closure, because we know birds move around, they have little wings, even feet, and it's just really hard to imagine that they are going to stay put, especially if the count duration is longer, or just over multiple visits, nothing is going to happen to them. Other assumptions might include that the observers are independent, all individuals are identical with respect to how often they vocalize or move around, their spatial distribution is random, and so on. We're going to inspect these a lot more and just see which assumptions are most important to be aware of and which are the ones that we can safely ignore. So just think about that. We know there is relativistic time dilation, but we usually don't care about this unless we want to synchronize satellites on a global scale, or we are on a space station. So usually for us doing field work, this is negligible, although it's a fact that it exists. And samples are independent. This is another assumption that we know because we've learned about it, but we usually try to not think about it that much when we are actually doing data analysis. And we just think, oh, let's throw in random effects or something like that will take care of non-independence that we know exists because we can't really achieve fully random sample over large areas. So all of these assumptions are there, and we are going to see there's a lot more. Let's just skip this for now. So the question we are asking really is, does it matter in practice if we have an assumption and we know that we are violating it to some extent? And one reason for this is because we want a simpler model. We seek simplicity, and also this just dictates whatever doesn't really matter. We should simplify that part of the model and focus on where really the problems are. So we're going to introduce a concept, understand how we can take some field collected data and estimate a parameter related to that concept. So if we are looking for abundance as our interest, then how we can estimate something so that we can get that metric that is going to tell us what abundance is, and maybe we'll have to estimate a couple of other things along the way to account for detection error. So how we can infer that parameter of interest from the data. And to better understand what is really going on, and now it's COVID and it's still just the beginning of the spring, so we won't be able to go on a field trip and do it outside. So we're going to create situations in silico, and by that I mean on our computer in a safe environment, just do some simulation, look at how things change if we move some sliders or use different settings. And then we'll look at how the outcome changes and how that relates back to these assumptions and the models. So really, we have all these assumptions, and it's guaranteed that we violate all of these. And so how much is too much? We can get a sense by doing these realistic simulations and just thinking hard about the model. And this is just a quote from me. All assumptions are violated, but some are more than others. I'll have a few puns like this on the upcoming slides. So this is really how I approach these 4 days, that we have at the end a very good understanding of what is really important to consider and what is it that we shouldn't care too much about. So now let's talk a little bit about bird data sets. So if you look at help pages of our functions or just tutorials over the net, you're going to see the iris and empty cars data set being used, but real data is often more ugly. I love gremlins, and that's why this little image is here, kind of showing the contrast between these example data sets versus the real data. And we are going to use real data sets here, but I already pre-processed it to some extent, so we are not going to see the full gremlin side of it. And this data set happened to be in a really good shape to begin with, so it was more Mogwai-ish. And in a tidy workflow, probably this is all familiar to you if you are using R. This is from the book of Hadley Wickham, R for Data Science, and Gary Krolemon, I think the other author is. So you take some data within your program. So all the fieldwork was done outside of this box. You have the data, you import it, tidy, transform, visualize, model, you go around a few times, and then you try to communicate it, you give a talk, you publish a paper. And this part is when you understand the data. So this is the workflow. But what I found is if you include time tracking, how much time you spend in each of these steps, then you're really spending 60, 80% of the time with tidying up the data. And then the rest is the more enjoyable part. So what biologists enjoy most is either outside of this box, they go out to the field, collect the data, or some of us likes these visualize, model cycle, and also the communication part. It's a great feeling when you give a talk and people ask questions or just get that paper out of the door. But still we spend lots of time tidying the data, and that's why I decided to spend some time on this. So that part is often called data processing, data munging, wrangling, cleaning, but really we should find some word to kind of bring through this dread that I often feel when I again run into the issues and just see error after error, and I have to spend days just cleaning up the data. So all data are messy, but some are missing. So this is another quote from me based on my experience with data. But this is really not a surprise. I call this the first mover disadvantage. So if you are the first one looking at the data that either you collected and the table is still a mess, or someone else did that, obviously you are going to be the first person to find those errors, and it's up to you to fix it. So that's why having some R skills is going to help you along the way. So if no one looked, no one found the problems yet. And I can mention very few data sets that I looked at and I found no issues with it. And that's just because I was the first one to do so. So whenever there's an update in the boreal avian modeling project dataset, or the Alberta Biodiversity Monitoring Institute, another organization I work for, there are always issues, and then this cycle begins of fixing those issues and making sure that the data set others are going to access after me are nice and tidy. So the other good thing about data is there are very few things that can go wrong. It's usually you get the wrong coordinates, or you have the wrong time, or you have the wrong species, or something else. But usually the first three happens. You have mis-IDs, or just you have a label that doesn't make sense, or you use the wrong zone, or just have a sign missing, or the time is not in a standardized format. So these are not very easy to deal with, but manageable, but kind of just comes down to the three axes of how we look at the data: where it is, when was it collected, and what species. So these are all very important when we then try to make sense of the data. So a little bit about the data set that we are going to use, or the first data set that we are going to use. This was collected under the Joint Oil Sands Monitoring Program, and this is an area within Alberta. This is Fort McMurray, and the green shading indicates the oil sands monitoring program. And then the dots are just the point count locations. So this is where the data is coming from. It's quite a large area. And then inside that, there were existing point counts, and those places were not always revisited, but then some complementary sites were found around those, so that we have a good balance spatially and with respect to land cover types, and also a nice gradient of all kinds of human disturbance, because in this landscape you get forestry and all kinds of energy features, to some extent agriculture, being in the same landscape. So we're going to have some of these landscape variables in our data set so that we can relate the bird point counts back to these. Just to give you an idea about this really patchy environment. So when we design our sampling and we want to replicate that, what we have in mind is that we want to make inferences about some aspect of the population, as we've seen before. And the full census, kind of going out and search everywhere, count everything, is out of reach. So that's why we need to take a sample of this population. This sample needs to be representative so that we can talk about the whole area other than just those places that we've visited. And having a random sample is useful because we can't really control for everything, and that just helps make those uncontrolled variables easier to deal with. Sample size should be as large as possible. When I was doing my PhD, I thought a couple of hundred or maybe 1,000 is a large sample. Now I'm looking at hundreds of thousands of points, and even sometimes millions. So this is relative. If you are starting out, maybe 100 is large enough because you know that effort that went into collecting all those data. But usually under 20 is considered something really small. And we will see that sometimes you need a couple of hundreds or at least 50, 70 detections to really talk about some of these detectability aspects of the data. So sample size can be limiting. But if you have more samples, that reduces variability and increases statistical power. So that's something good to have or think about early on, how to maximize sample size within constraints. And then when we need to pick where to go, which survey locations to choose, then we often do stratification, maybe based on access or land cover types. We're identifying gradients that we want to study, so that we are not just having 0 and 100% of those features, but maybe something in between, so that we can fill in all those gaps. And having randomly chosen locations helpful in controlling for unmeasured effects. And then also, if there are historical surveys in the area you are going to, you might decide to avoid these, so you have more independent locations to use. Or if your question is more about trend, then you might decide to revisit these sites so that you can talk about how things have changed over time.